Hello student, Namaskar. I am Dr. Sam Patel, Assistant Professor in ITM University Gwalior. So in BSc AG course, as you are in 5th semester, that is third year, and I will teach you plant pathology subject. And in PP 311, the course title is Diseases of Field and Horticultural Crop and Air Management First, in which only study field crop diseases. So today I will start the unit number 5, that is diseases of gram, that comes under lecture number 27. So first I will give outline of the today lecture, introduction to gram diseases, and what are the different objective of the today lecture, then what are the economic importance of the today lecture, why we need to study these uh, gram diseases, and in gram today I include only 4 diseases. First one is rust, second one is a dry root rot, and the third one is a wilt, and the last one is a Ascochyta blight. After completion of the diseases, I will give one exercise, and I also explain what are the learning outcome of the today lecture, then what are the different research scope of the today lecture, and at last I quoted the reference. So these are the outline of the today lecture. Then first, introduction to gram disease. As you know that gram is a very most important pulse crop in India, and in pulse gram has highest area and production in India. So most of the pulse demand fulfilled by the these gram crop. And as you know that in India there is a shortage of the oil seed and pulse. So most of the demand of the people fulfilled by the importing these pulse and all seed crop from the outsource, so that means foreign country. So government of India has given a more emphasis to increase the area and production of the these pulse and oil seed crop, and gram is one of the most important crop or pulse crop. So and gram crop is cultivated in Rabi season, and because it is cultivated in Rabi season, so more it is prone to more attacked by the different type of insect and pathogens, and mostly rust, dry root rot, wilt, and ascochyta blight is most these diseases causes huge losses on the gram crop. Then what are the different objective of the total lecture? So to study the various disease of the gram, in which I explain, I cover only 4 diseases, that is wilt, rust, and other thing. Then detection and identification of the pathogen, in which I explain the what causal organism was responsible for causing large number of diseases in the gram, and how we detect these pathogens in the gram crop in the field. And to study the disease cycle, in this I explain the what are the primary source of the inoculum, what are the secondary source of the inoculum, and how these pathogens completed their disease cycle in the host. And last one is to study the management of various diseases, in which how we manage or control the diseases in the field condition, and by controlling or managing the diseases we increase the production of the gram crop. Then economic importance of the today lecture. So as you know that a large number of disease occurs in the gram crop, and almost 25 to 30% crop losses due to the gram diseases. If farmers control these diseases in the gram in the field, so by controlling the diseases they increase the production of the gram crop. When they get more yield, then surely they also earn more money, and they increase their life standard. So controlling of the these gram diseases is most important to the farmer. Then first disease is the rust, and the rust in a gram is caused by the Euromyces ciceris ariantini. These are the most important diseases in the gram, and almost 10 to 20, 15% crop losses due to these rust diseases. And these rust diseases are most prevalent to Madhya Pradesh, Andhra Pradesh, Maharashtra, and Uttar Pradesh, where gram are cultivated. And mostly these diseases are prevalent in Madhya Pradesh. Then in symptoms part, the infection appears as a small oval brown powdery lesion on the both surface of the leaf. As you know that if we consider the typical rust symptoms, so orange or brown color pustule appears on the firstly lower surface of the leaf. When severe condition is occurred, then these pustule present on the upper surface of the leaf as well as stem and the flowering parts. And so first infection appear as a small oval brown powdery lesion on the both surface of leaf, especially on the lower surface, and these lesions which are iridosaurai cover the entire leaf surface, and sometimes a ring of the small pustule can be seen around a larger pustule which occurs on the both leaf surface. Later in the season, dark teleosauri appears on the leaves, and the rust pustule may appear on petioles, stem, and the pods. And it is heterogeneous rust, for completion of their life cycle, they require another collateral and alternate host for completion of their life cycle. So in gram, these heterogeneous rust discovered, but the pineal and axial stage are unknown. So as you see in the photograph, there is a orange or brown color pustule appear on the lower surface of leaf in the photograph of this disease symptom. When severe condition is occurred, then these pustule also present on the upper surface of the leaf and as well as stem. So these are the symptoms of the rust in the ground. Then what are the pathogen characteristics? So the iridospore are spherical, brownish yellow in color, loosely attenuated with 4 to 8 germpores, and these teleospore are round to oval brown, single-celled with thickened apex, and the wall are rough brown and warty. So these are the pathogen characteristic of the Euromyces caesari serentini. Then in disease cycle path, the fungus survive as iridospore in the legume weed such as Trigonella or Polycerata during the summer month, and serve as a primary source of the infection, and the spread is through by windborne iridospore because these disease spread with the help of the air. So within a very few days it cover the entire field. In management part, if we control the disease weed host such as Trigonella polycerata, then we control the primary source of the inoculum. So main crop was not infected, and if we dust sulfur as 20 kg per hectare or spray mancozeb at the rate of 0.25%, then we control the disease. And if we give seed treatment with the mancozeb at the rate of 0.1% and or Pantavax at the rate of 0.3%, then by this seed treatment we also control these rust disease in the gram. The next disease is the stem and root rot or dry root rot disease in the gram, and this stem and dry root rot disease caused by the Rhizoctonia bataticola, and the pineal stage of the Rhizoctonia bataticola is Macrophomina phaeolina, and the sexual stage is Thanatophorus cucumeris. And as you know that as I told earlier, you remember these perfect are a sexual stage of the Rhizoctonia, that is Thanatophorus. These are the very important, and many times question was asked by these sexual or perfect stage of the Rhizoctonia. And this disease is the most important because this disease mainly attacked on the seedling stage. So if a seedling of the gram is attacked, then we see the seedling of the gram is die or dried. So by drying of the seedling, the plant population of the gram is decreases in the field. In symptoms part, the disease generally appears around flowering and podding time in the form of the scattered dried plant, and seedling can also get infected. The first symptom of the disease is yellowing of the leaf, and affected leaves, petioles, leaflet drop within a day or two, and the leaves or stem of the affected plants turn extra color and plant build within the week. The lower portion of the taproot usually remain in the soil when plants are uprooted. The taproot is dark, showing symptoms of root rotting, and is devoid of the most of the lateral and finer roots. Dark minute sclerotial body we can also see in the root, and these sclerotial body exposed in the wood. As you see in the photographs, the clearly we see the root rotting of the root of the gram, and we see sclerotial body of the fungus in the root as a black color. And so these diseases are very important, and mostly as the name indicated, root rot or dry root rot, so mainly root are affected. So if root are affected, then translocation of the water is also reduces, and by deficiency of the water in the upper portion of the plant, so sometimes a plant are died because of the shortage of the water. Then in pathogen characteristic, the hyphae of the fungus are dark brown, filamentous or septate with a constriction in the hyphal branch at the junction within main hypha. These are the most characteristic of the rhizoctonia, and the sclerotia are brown or irregular in shape because these rhizoctonia form a sclerotial body, and this sclerotial body has a round shape, thick walled, and look like a mustard seed. And sclerotial body serve as a dormant stage for long period. Whenever favorable condition or comes back or return back, then these sclerotial body germinate and form new fungus, and these new fungus cause or affected the new crop of the gram. And the fungus has its sexual stage. It is a Theratophorus cumaris, which produce 2 to 4 basidiospore in terminal cluster of the on the hyphae. And in disease cycle parts, the pathogen survive in the soil in the infected host debris as sclerotial body for several years, and the sclerotial body serve as a primary source of the inoculum, and the secondary spread is through the farm implements, irrigation water, as well as rain splash. So these are the disease cycle of the Rhizoctonia bataticola. And in favorable condition, if maximum ambient temperature above 30 degrees centigrade and minimum temperature is 20 degrees centigrade, and moisture stress condition is there, then these conditions are very much favorable to development of the dry root rot or root rot disease in the gram. And in management part, if we give seed treatment with a carbendazim or thiram at the rate of 2 gram per kg, or seed pelleting with a trichoderma viridi at the rate of 4 gram per kg, or pseudomonas fluorescens at the rate of 10 gram per kg. By these we control the diseases, and we also control the diseases by growing tolerance genotype such as ICCV10. These are the tolerant genotype of the gram against the dry root rot diseases. If we apply FYM, that is farmyard manure, at 10 ton per hectare, then we control the diseases. The next disease is the wilt disease in the gram, and this wilt disease is caused by the Fusarium oxyscorum pharma species ICRI. Wilt disease are most important in the gram crop because in wilt disease are most it occurs all growing area of the or cultivated area of the gram in the India as well as in the tropical and subtropical country, and yield reduces up to 30% when severe condition is occurred. And mostly these diseases are prevalent in the Madhya Pradesh, Uttar Pradesh, and Rajasthan. And in symptoms, the disease occurs as 2 stages of the crop growth. First one is a seedling stage, and second one is a flowering stage or adult stage. The field symptoms of the wilt are death of the seedling or adult plant in the patches. So because the seedling and adult plant are died because of the wilt disease, so plant population of the gram is decreases in the field, and we clearly see in the field as a patches in the field available in the because of the wilt diseases. The seedling collapse and lie flat on the ground, retaining their dull green color, and when split open or cut transversely, brown to black discoloration of the internal tissue can be seen. Because of the mainly build condition or occurs because of the disturbance or discoloration of the vascular tissue. Because so if vascular tissue are damaged, then translocation of water is restricted. Because translocation of the water is restricted, then plant tissue building symptoms. Grown up plant is a typical symptom of the wilting, that is dropping of the petiole, rachis and the leaflets. All the leaves turn yellow or then light brown, and vascular discoloration is observed on longitudinal splitting of the stem. Sometimes only a few branches are affected and resulting in partial wilting. As you see in the photograph, clearly we see the drying of the plant because of the wilted diseases. Then in pathogen characteristic, the fungus that is Fusarium have a hyaline to light brown septape and perfused around hyphae, and these Fusarium forms microconidia as well as microconidia. Microconidia are oval to cylindrical, hyaline, single-celled, normally arise on the short conidiophore, and microconidia which burn on branches conidiophore are thin-walled, 3 to 5 septate, fusoid and pointed at both ends. Chlamydospore also produced by the Fusarium, and these kind of chlamydospore are rough-walled and smooth terminal to intercalary, and maybe is singly or in a pair in a chain. So these are the pathogen characteristic of the Fusarium, and the most important pathogen characteristic is these Fusarium produce micro as well as microconidia. In disease cycle part, the fungus may be seed borne and survive in the infected plant debris in the soil. So seed and as well as infected plant debris that are present on the soil serve as the primary source of the inoculum with the help of the chlamydospore that present on the soil. And these chlamydospore remain viable up to next crop season, and the weed host also serve as the source of the inoculum. The secondary spread is through the irrigation water, cultural operation as well as implements. So these are the disease cycle of the wilt disease of the gram. And in favorable condition, if soil temperature above 25 degrees centigrade, high soil moisture, monocropping will occur, and if present of the weed host such as Cyprus rotundus, Tribulus terrestris and Calbulus arvensis. So these weed hosts also serve as the source of the inoculum. So these are the favorable condition for development of the these wilt disease in the gram. And in management part, if we give seed treatment with carbendazim or thyram at the rate of 2 gram per kg, or we treat the seed by trichoderma viride at the rate of 4 gram per kg, or pseudomonas fluorescens at the rate of 10 gram per kg, then we control the diseases. And if we apply heavy dose of the organic manure or green manure in the field, by this application of the green manure or organic manure, then we decrease the inoculum that present on the soil. And if we follow 6-year crop rotation with non-host crop and grow resistant cultivars such as Kranti, Sweta, Avrodhi, Pusa 212 and JG315, these are the resistant cultivar against the wilt disease in the gram. Then next disease is the Ascochyta blight, and Ascochyta blight is caused by the Ascochyta arabi, and perfect stage is Mycosphaerella pinoides. These are the most important diseases in the gram, and almost 10 to 15% crop loss due to the dead disease. And dead diseases are mostly prevalent in the Maharashtra and the Madhya Pradesh region, and in these two states almost in every year the disease are present. And in symptoms for the disease generally appears around flowering or podding time in the form of the scattered dried plant. As you see in the photograph, there is a yellowing symptoms present because of the Ascochyta blight, and these symptoms present in the all above ground part are affected by the plant, and the disease is usually seen around flowering and potting time as patches of blighted plant in the field, and on leaves small water soaked necrotic spot appear on the leaves, and these spots are enlarged rapidly whenever favorable conditions are available, and leads to blighted symptom. Pycnidia are also observed on the blighted part, and in hot dry weather the infection remain in the form of the discrete lesion on the leaves, stem, pods, and seeds. On leaflet, the lesions are round or elongated with a grey center surrounded by brownish margin, and similar spot may appear on the stem and pods. The spot on the stem and pods have pycnidia arranged in concentric circle as minute black dot. The stem and petiole usually break at the point of the infection due to girdling, and if the main stem is girdled at the collar region, the whole plant die, and these seriously reduce the yield of these gram crop when Ascochyta blight disease occurs. So main symptom of the Ascochyta blight is the brownish spot appear on the leaves, and these brownish spot are enlarged and form a blighted symptom. So and sometimes yellowing of the leaves also occur due to the blighted symptom as you see in the photograph. Then in pathogen characteristic, the fungus produce hyaline or to brown septate mycelium, and pycnidia are spherical to subglobose with a prominent ostiole. Conidia are born on short conidiophore inside the pycnidia, and they are hyaline, oval to oblong, straight or slightly curved, and single-celled. Occasionally they are biocelled. The perfect or perithecial stage is also seen in the infected host tissue, usually after the plant is died, and the perithecia are globose, dark colored, and conidia, and these perithecia also contain acai, which are typically 8 spore, and these ascospore are hyaline, thin-walled, and two-celled. These are the pathogen characteristic of the Mycosphaerella rabi, and then in a favorable condition, if night temperature is 10 degrees centigrade and day temperature up to 20 degrees centigrade, and rain accompanied by the cloudy weather and excessive canopy favor the disease spread. And in disease cycle part, these fungus survive in the infected plant debris as a pycnidial stage, and the pathogen is also externally and internally seed-borne. So pycnidial that present on the infected plant debris, and seed serve as the primary source of the inoculum, and the secondary spread is mainly through the airborne conidia. Rain displays also help in the spread of the disease. In management part, if we grow resistant or tolerant cultivars such as Gauro C235, G543, GG588, GG688, BG261, and GNJ214. So by these by cultivation of these resistant or tolerant varieties, then we control the Ascochyta blight diseases. In cultural management practices, if we remove and destroy the infected plant debris in the field and follow crop rotation with the cereal, if we sowing deep sowing of the seed occur, that means below the 25 centimeter, and intercropping with the wheat, barley, and mustard. By this cultural operation, we control the these Ascochyta blight disease. And if we give seed treatment with Thyram 2 gram per kg or carbendazim at the rate of 2 gram per kg, then we control the diseases. We also control the disease by exposure of the seed at 40 to 50 degrees centigrade, reduce the survival of the Ascochyta rabi by about 40 to 70%. And if we give spray at the rate at carbendazim at the rate of 0.1% or chlorothalonine at the rate of 3%, 0.3%, then we control the Ascochyta blight in the gram. So in today's lecture I cover the 4 diseases, that is wilt, rust, Ascochyta blight and dry root rot. If you have any doubt, then you call me or contact me. Now I will give exercise to you. So you go to the gram field and collect the symptoms of the each diseases on the basis of the pathogen and symptoms, and you also when you return back to the college, then you at time you prepare a slide and you also isolate these pathogen, and you also expert to identify the diseases on the basis of the symptoms. Then in learning outcome part, I explain the 4 diseases such in the that is wilt, rust, Ascochyta blight and dry root rot or stem rot. And each disease I explain the what are the causal organism, what are the symptoms, economic importance, pathogen characteristic, favorable conditions, disease cycle and management. I also explain the how you identify the diseases in the field conditions. So these are the learning outcome of the total lecture. Then you also perform research in the on the basis of the gram diseases. So first one is to develop eco-friendly management of the diseases. In this you search best strain of the trichoderma and pseudomonas for controlling the fungal as well as bacterial pathogen. You also perform your research such as survey and screening of the gram disease in the Chambal region, and after surveying you collect sample from the different region of the Chambal near the Gwalior, and you isolate these samples in the lab, and you suggest which isolate is the most virulent for causing diseases in the particular region as compared to the other region. And you also perform a research as a best for selection of the best botanical extract for controlling the this pathogen on the basis of the poison food technique. So these are the research scope part of the total lecture. Then these are the references from which I make the presentation, and if you want to further study, then you go through these books, and you also search ICRE course in the website. So with the so I completed 4 diseases in the gram, and if you have any doubt or after seeing my video, then you call me or contact me. I will try to solve your problem. You also ask your doubt in the webinar class. And with this remark, I concluded to the lecture. So thank you for watching my video. Thank you. Best of luck.